how we're going to be planning for our egg lab, the things that you should be thinking about, and how you can complete the next assignment on your own. So just a reminder, we're learning about calorimetry, or the study of energy transfer, and we've learned this equation QMAT. Q being the amount of calories needed or transferred in a system to make something change. In this example, we're going to be heating a pan, which will be heating oil, which will be cooking an egg. The mass is the mass of the substance in question. We could be measuring the mass of our pan, or the oil, or the egg before and after we cook it. A is, again, the specific heat, really the reactivity of a type of material. The standard bearer of A is always 1 calorie per 1 gram per 1 Celsius degree change for water. If you see that number higher than 1, it means it's very slow to react. If you see a very small number, it means it heats up quite quickly. And then finally, T is the change in temperature. There's a video here, again, showing you how to calculate mathematically QMAT for different types of examples. Now, this is important because specific heat is the measurement of how something changes when it is given energy through some sort of system. And specific heat really refers to how quickly a change occurs within a system. We're going to be testing a variety of things in our egg lab based on solids versus liquids, pans versus oils and eggs, metals, which have a really low specific heat, they're quite reactive, and then complex molecules such as eggs that tend to take more energy to heat. Now, temperature is the measurement of movement, but temperature is also the measurement of how well our egg is going to be cooked. Now, hopefully you feel good about your egg frying lab and the basic setup. The idea is that we're going to be testing different types of pans, different types of oils, and how that influences the final cooked fried egg. We're not doing scrambled or omelets, we're just doing a sunny side up egg with a firm yolk and a finished white. So today's work is going to be looking at an experimental planner where I'm going to be assessing each of your ability to plan an investigation. It might not be the investigation that I actually do at home to collect data, but I want to see that you're able to plan an experiment based on these ideas. We have 3 main objectives. You can write a specific question regarding the lab at hand. You can propose specific treatments to answer your question that you propose. And you finally explain the types of data that you'll collect to best answer the question. So the first thing that you're going to be doing today is defining a scientific question. We have different types of independent and dependent variables related to QMAT, and there might be other variables I didn't list. You want to write your specific scientific question, which might change as you complete this document, your independent variable, and then finally the dependent variable. You can have more than one. As you change the independent variable, types of oils, types of pans, types of metals, your dependent variable, final temperature of the egg, time to cook the egg, will change. A good example of this is, does the length of a person's leg determine their vertical jump height? So going on to part 2, you're going to be planning out different treatments to test your question. We need to have enough data to make sure that we can answer our question confidently. In this table here, you're going to be proposing the different independent variables that you will measure to see change in your dependent variable, i.e. you could add more columns if you have more than one dependent variable. And then I want to see a brief explanation as to why you chose this variable or treatment. So going back to this question, does the length of a person's leg determine their vertical jump height? My first independent variable might be 5 people with 36-inch legs. I want to see how tall people have a vertical height. I might do the same thing with 5 people with 30-inch legs. And then 5 people with 24-inch legs. You're really just proposing things that you think would be appropriate to answer your question. Make sure after you do this, you choose one of your treatments as the control. The control treatment is the best attempt at establishing a baseline or an average environment. So the average length of people's legs is 30 inches, so that would be my control treatment. Now the final thing that you're going to be doing is proposing the way in which you're going to collect data, making sure that you maintain accuracy and precise data between trials. So if I was going to measure people's leg height, I might measure from midfoot metacarpal, which is the big bones in your feet, to the hip bone in the center of the outer hip. I'm going to be being really specific with how I measure my variable in order to make sure I do it the same way each time. And that's how you're going to be completing your experimental planner for the egg lab.